welcome everyone. Great to have you actually here in person and something like 60 people online. So it's a typical New World meeting. This is the first of two partnership seminars that some of our colleagues from two of the large UQ-based research centres, research institutes, are going to present to us, really to support linkages with the clinic, but also with the research excellence we have within TRI. Unfortunately, Alan Rowan wasn't able to come, but we've got an even better speaker now in Professor Trent Munro. Trent is a biotechnology and biopharmaceutical professional with extensive experience in the development of a wide range of biologics. He's currently Senior Group Leader at AIBN, the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland. He's also the Director of the NCRIS-funded National Biologics Facility and Program Director for the CEPI-funded Vaccine Rapid Response Pipeline, and really had a major role with his colleagues at UQ in the UQ COVID-19 vaccine development. It's great that you're able to come in person today, Trent, and he's going to speak to us about bench to commercial manufacturing, something that's key to the discussions in and around TRI at the moment. So welcome, Trent, back to TRI. Great. We look forward to hearing from you today.
Thanks, Scott, for the kind introduction, and thanks for the opportunity to come. Alan does send his apologies. So I'm going to split this talk up a little bit and spend a little bit of time talking about AIBN, because I think not everyone might be familiar with current state of where AIBN is at and some of the focus areas and strategy there. I'm then going to talk a little bit around, my views on what's important when we're thinking about developing a product. And when I talk about developing a product, I'm primarily talking about the manufacturing paradigm. So I'm going to talk about that. And then if we have time, I'll just give a little bit of an update on some of the latest on our vaccine program. I know Paul Young has been over and given a more detailed seminar on a lot of the data. But I'm just going to talk to you a little bit about strategy and where we are there and what the lessons learned from that exercise were around how we think about translating therapeutics. So just to start, because I realize that St. Lucia is just across the river, but sometimes it seems like even that's a bit of a distance. Just going to get these slides moving. Okay. So for those who don't know, AIBN is over on St. Lucia campus. It opened in 2006. It was one of the number of institutes around Brisbane that, like TRI, benefited from funding from Atlantic Philanthropies to leverage other government funding. It's an interesting place in terms of that the focus is quite wide and multidisciplinary research areas. It's home to 27 research groups, and they work across the entire continuum of biological, chemical, and physical sciences. And more recently, they've focused down on thinking about, it used to be 5, but now 3 major research themes, and I'll tell you about those. Advanced biomanufacturing, which is obviously the one that I'm most interested in, precision nanomedicine, which is an interaction area for me, as well as advanced materials. And so before I start, I did just want to say that I have a bit of an interesting background and came back from industry working in the US in late 2019, pre-COVID, not knowing what was going to unfold. And as part of that role, joining UQ, took on a variety of different roles. So I'm not a traditional research academic, I would say, if that thing still exists. But really am focused on driving outcomes for researchers, and the primary outcomes I'm interested in is really driving products out of impact-driven research. And so roles that Scott mentioned, I'm a project director on the CEPI-funded Rapid Response Vaccine Pipeline. I do a little bit of work in helping state government with some of their biomanufacturing strategy. I run an NCRIS facility that's really, I'm going to spend a little bit of time talking about it, that supports ability to develop protein-based biologics. And we've recently brought online an RNA facility there that I'll talk about. and I'm also the director for an ARC training center in biopharmaceutical innovation. You can see some of our partners there, and I won't spend any time talking about that, but happy to take questions. When we think about AIBN as an institute, it's got many, I guess, parallels to what's here at TRI. One of the things that really underpins it as an institute, and is something that's, both, I think, one of its strengths, but also potentially one of the challenges we have in terms of outward communication, is we have a large number of core facilities within the building. And I think of these core facilities as really some of the most important things that researchers can access. So they don't have to maintain equipment. They don't have to worry about getting very large grants. They don't have to worry about upgrading equipment, that they can come and access freely within the AIBN. And a number of these are funded under the NCRIS framework. And for those who are not familiar, that's the National Collaborative Research Infrastructure Strategy. And across that, we have nanofabrication facility, a metabolomics and proteomics facility, microscopy and microanalysis. We have a data center, and we also have a stem cell center, as well as our own biologics facility. So you can see all of those hexagons there around the NCRIS framework. These are all nodes of part of national frameworks as well. And I'll talk about our own one on that. Beyond that, there's aims to develop an organoid facility. And we've recently received a couple of $1 million in funding to boost our ability to do nucleic acid-based translational research. Also, we're looking at branding some of this under a biomanufacturing center called ACAB. You'll hopefully hear more about that into the future. And we also have other core facilities, like a large PC3 facility that's currently under construction, Linda Lewis's protein expression facility we work very closely with. There's a very large animal house like there is here, and there's high performance computing. And all of these things fall under sort of thematic areas like nanofabrication or metabolic engineering, dealing with proteins, or dealing with how we engineer systems and organisms. So for those who are not familiar, please reach out if any of these strike a chord, and I can make sure we put you in touch with the right people. In terms of the size of what we are in terms of people, there's around 400 people in the building. I should say I joined AIBN just as it was getting built, just before it was getting built. I think one of the first employees back in 2006. And so it's been very interesting to both have seen it go through initial phase of growth, be away for a while, and come back now and seeing where the Institute is at. There's more than 400 people in the building. Depending on how you count it, between 27 and 29 research group leaders, large number of postdocs, more than 100, and now up to 160 HDR students as well. Then you can see around that all the different mechanisms that those people have of the way that they exist within the building. And I think, a credit to AIBN is the fact that there's 3 ARC laureate fellows in the building, which is something that's a real boost for the research outputs from there. Okay, so this is not the area that I would usually talk about. This is Alan's area to try and sell the Institute, but it's also very important to think about how do we get our income, what sort of outputs do we have, and how is the Institute rated? And I think this is especially important at the moment across the board. And I'm going to talk about Brisbane potentially in terms of the number of research institutes we have and how they either work within a university system or work independently. But what's the value that research institutes bring working together with more traditional faculties and align with teaching needs of universities? And I think this is going to become a very important topic for us all to become aligned with and au fait with over the next little while. But suffice to say that since 2016, there's been quite a lot of growth in AIBN, a number of new group leaders, nearly a 30% increase in academic headcount, had 148 HDR students and 26 completions per year since that time. And you can see from a commencements point of view, that continues to go up. And on the next slide, I'll show you why that's also a challenge. And I'm sure it's a challenge here with the current state of borders, et cetera. In terms of income, vast majority of income is from Cat 1 based competitive grant income, but we also, of course, get a number of other diverse income sources across the board, as would be normal across most research institutes. And so one of the other things that, of course, is an important measure is thinking about expenditure and how we look at that income and really how that balances off against our total expenses. And I think the main thing to note here is that this is continuing to increase. We don't see any decline in this. What we do see, and I'm sure it's something you all struggle with, is how do we diversify that income across an ever broadening range of schemes, some of them very specific, some of them very broad, and then how do we continue to engage in the schemes that require matching from universities, things like CRCs, et cetera, which require internal support. And of course, the competition for internally backed grants is becoming increasingly difficult for people to deal with, because there's just so much pressure on the system. So we all know that now for many grants, they don't cover the true cost of research. We've really lulled ourselves into the situation where we've kind of accepted that even the tier one research income doesn't actually give us what we need, and that the universities are expected to find money from elsewhere to support that. And I don't think that's going to continue. I think we're in for some challenging times there. And the main reason for that, of course, is when we look at the breakdown of our students and the geographical origin of a lot of those students. And so for us at AIBN, mainland China is a very important source of our higher degree research students, as it is across the board. But this is particularly apparent for us, especially if we look across some of the areas of biotechnology and some of the other chemical areas, where really those students are the ones that are really interested in coming and doing that research, equally with Japan and South Korea. And so I think, while we've been able to sustain through the current COVID crisis, I don't think we're going to be able to keep that going too much longer. So we're really hoping that this situation changes and we can work to bring all of the students who've either got accepted places or are looking to come and study and start higher degree research here in Australia very soon. Okay, so with that, I'll just move on to how we came to these focus areas and what they mean. And so I think it's very difficult to try and define, when you're working in such a multidisciplinary environment, how you fit into a box. And so one of the discussions we've been having is that this is not meant to constrain the activities that happen within the Institute, but it's meant to give us a directional focus on how we think about the most impactful outputs for that research. And so the areas for AIBN, as I mentioned right at the start, are these intersecting circles of advanced biomanufacturing, precision nanomedicine, and advanced nanostructured materials. And within that, I think one of the areas of strength that's become, that's continued to grow at AIBN over the years has been the idea of clean energy. And what I mean by that is our ability to store different types of energy sources in novel ways, using elements across these three intersecting circles of this Venn diagram. So if we think about the split out of some of these things, if we think about what's in the precision nanomedicine bucket, it's things like diagnostics, drug delivery, theranostics, et cetera. If we think about the materials side, it's really both the synthesis and study of those nanomaterials, whether it's inorganic, hybrid, biomimetic, i.e., some part biological, some part synthesis. And it's also discovery science with broad application. So really thinking about natural materials or other manufactured materials and how they can be used in novel ways. In the advanced biomanufacturing sector, we're really connected to thinking about things along the synthetic and systems biology area. We've got our facilities that I mentioned earlier. We've got the stem cell areas. And really that's about applications that span health, environment, and sustainment. Parts that sort of sit within that are things like nano agriculture, things like stem care, provision of services for engineered stem cells, for example. And so all of those things come together. And as we've worked on this as a team at AIBN, one of the things that we've had to do as a group, which I think is quite challenging across such a diverse range, is look across all of the groups, look across all of the research that's going on, and look at saying, okay, which buckets do people fall into? Which areas of their research do they come into? So just, there's some names up here you'll see in the green boxes. These are the people that, some of these names might be familiar to you, some of them maybe not. These are the people who are working in these different areas, whether it's focused on diagnostics, people like Matt Trow or Andrew Whitaker, or people focused on drug delivery, like Alan or Michael Montiero and others, Tom Davis, people that you know, or people working in the theranostics area. I mean, many of you will know Chris Thorect and Andrew Whitaker again, who covers both of these areas. Think about the advanced biomanufacturing. Again, we've got these same thematic areas that I talked about. And I think the really important thing about the advanced biomanufacturing here is why I'm obviously very interested in the medical side of advanced biomanufacturing. That's my particular area, that we've got a range of people working across different types of advanced biomanufacturing. So that's coming up with what they call, Industry 4.0, really thinking about how we're going to change the way we make the building blocks, whether they're for energy, whether they're for high value goods for the chemical industry or other areas. How do we go about synthesizing those in different ways than we do today, primarily using highly engineered organisms to do that? And so, and underpinning that, of course, there's a huge opportunity in data management across these areas, because if we look at the scale, the omics revolution that we've all been living over the last 10, 20 years, I think the sort of precipice that we've got ourselves at is, how do we use the data coming from all that in ways that actually make sense? How do we filter out the noise and filter through to functional solutions? And so we've got groups there working on the systems biology and functional genetics underpinning that. Our connection points, our major touchbacks here to the faculties, obviously both with the engineering and SCMB school at UQ. And just to finish off on the third area of advanced functional materials, again, a wide range of centers that support this. But if we think about what's in there, we've split it up here into energy, functional materials, and this new idea of adaptive materials, things that may come across a number of different uses. And again, we're working with both the medical school on one side and engineering SCMB on the other. And again, the names here of the number of the group leaders who are working in this space. I will say that working in AIBN, the one real area of strength has been the interface, particularly on polymeric materials and biology. That's an area where I think there's a lot of very positive things have come out of AIBN, and an area that I'm particularly interested in, have benefited from a collaboration point of view. All right. So I'm just going to finish on one other thing that's happening at AIBN, a new scheme that I think we're particularly interested in, would be very happy to talk more about. I'm just going to give one slide on that. And that's an idea of how do we change the types of PhD graduates that we have. And so we've got a pilot program happening now, which is an entrepreneurial PhD program. And I'm sure we'd be very interested in collaborating with folks at TRI on this. If there's PhD students that you know of who might benefit from such a model, please come and talk to us. The way that we're really thinking about this working is that it's in addition to your PhD research project. So, this really is going to give a broader set of skills to folks who are interested both in the research side, but also in understanding how to use that research for different types of outcomes. And so the quality that I think we need to boost in Australia is definitely that entrepreneurship, that risk-taking, that idea of understanding how to drive something from just something you're doing on your bench to something that might become a product one day. And I think, we all recognize we've got the research quality here, but this is one attempt, a pilot attempt, to try and see how we can boost that. And that'll be a fairly complex scheme of ensuring that it's a point-based type of graduation or progress system. And that'll really be divided up amongst these different areas. There's sessions, experience, placement. So it's going to be a challenge. It's going to be a tough program, I think, to really make successful, but something that we're excited to get off the ground. All right. Okay, so that's AIBN. So I'm going to switch gears now. And, I always think it's great to be here. I love the name of this institute. It's definitely one of my favorite because it really sets the tone and intent of what's happening. And I think we've just seen so many amazing things as a result of this institute and others. But I really wanted to take a step back and think about what does it really mean to successfully translate research? I think that's a really hard question to answer, and I'm probably not going to answer it to anyone's satisfaction, but I'm going to give some ideas. And funnily enough, just on the bus on the way over here, even in the 5 minutes from St. Lucia, this popped up. And so I don't know if people have had a chance to see this in the latest issue of Nature Drug Discovery, but it's written by one of the NIH facility, a guy who works in one of the NIH facilities, which is very similar to our NCRIS framework. And it talks about translational misconceptions. And I just put this up here. You can look in Nature Drug Discovery and pull it up. But I think the questions here are really important ones. And I know I'm going to be sending it around to my team and around to the Institute, because I think it really, this is something we should be telling all of our politicians and all of our funders that it's not about, whether you fund late stage research or early stage research. It's about how you fund ideas and those ideas and how they flow into becoming important solutions for us. And it's not a simple journey. So just wanted to put that up. I literally pasted it at the last minute. All right, so practicalities. So what are the issues? Well, the big issue is always money, right? And so I think the really scary thing is that while we've made huge strides in the pharma industry over the last little while and lots of new products, that's been at a cost. And the cost is that it now costs well over $2 billion typically to take a product to market. And so I could spend, a whole day talking about why that's a problem. But realistically, I think it's pretty obvious that we do a really bad job of going from many candidates down to one candidate that makes it all the way through. And so I think we all need to be thinking about why that's the case. And I don't think, just giving the answer, well, biology is hard, is something that we can continue to use to make that acceptable. And I also think that it means that long-term sustainment of the pharma industry is at risk. One of the issues that we have is that the sector, the whole pharma sector, is actually really bad at translating spending in R&D through to outcomes when we benchmark ourselves across other industries. So if you look at in the pharma industry, in the blue bar across the top, you look at the amount of money that's spent in R&D, per head count or per return or whatever the measure is, compared to say communications or semiconductors or aerospace, and we're really just not in the same ballpark. So all of those sectors have matured. They all understand what it takes to have a long-term sustainable business, I think. And they've all adapted or been through crises to force that adaptation. Example being the motor vehicle industry, which has gone through, huge change in manufacturing strategies, geographies, et cetera. But they've had to adapt to bring that cost way down. You look across manufacturing in general, the cost is much lower, 10 times lower than it is in pharma, which pharma really is about manufacturing at the end of the day, because that's how costs are generally set. And what we end up trying to do is recoup our R&D spend. And you can see that in the table here. And this is what gives all the CEOs of pharma industries nightmares, because they've been living the good life for a long time, working in an industry where essentially you just make a lot of profit, so it doesn't really matter, to now where they're actually seeing their proportion of sales percentage being hit by how much they have to spend on R&D, and in a trend that's not going the right direction. So this, given our knowledge, given how far we've come, given we've gone through a data revolution, this really should be heading the other direction, and it's not. The good news is the types of molecules we're bringing forward is diversifying. So if you think about a modality, and I call a modality, a type of drug, if you look across, this is just FDA approvals, since 1995, you went from a situation where essentially the vast majority of approvals were these blue here, which is just small molecule drugs, through to a situation where you've really got a diversity of different types of products. And so that's good. We've got better at thinking about different ways of attacking biology, but we haven't really got better at making that cost effective. And one of the reasons is that it is hard. We've gone from making things synthetically, where we can characterize every atom in a structure, to making things that have very high molecular weights and are very hard to characterize and produce, manufacture. And now we've taken this even the next step when we think about making things like viruses or cell therapies, which are just incredibly complex. And so we know we've got a way to go there to really change that equation around, but something we all need to be focused on. I think one of the things we can do to focus on that is making better decisions earlier. But so why do we keep doing it? I'm telling you sort of doom and gloom stories. Why do we keep doing it? Well, the reason is because when you get it right, kind of makes a big difference. So this example I love showing, it's one of the first monoclonal antibodies that was commercialized. It's a pretty awful molecule, has a really pretty terrible history. It's not even the best therapeutic out there, but it's helped millions and millions of people. And it just so happens it's also made an incredible amount of money. So before biosimilar erosion, which is starting now for Humira, it was approaching $20 billion a year in sales. So lifetime sales of over $140 billion. So you're going to accept a lot of risk to think about developing the next Humira. And people are saying now, if you look at antibodies that are checkpoint inhibitors, for example, the PD-1, PD-L ones, they're developing huge amounts of revenue as well. So there's still a major revenue driver for innovation. All right, so this really gets to what I want to focus on, and that's getting it right from the start. What do I even mean by that? what does that actually, what sort of things should I be thinking about? Well, there's many things you need to think about on the journey of taking your research idea through testing and then into something that's actually going to be packaged and come as a commercial product. For me, one of the areas that we don't think about very much in Australia is how we manufacture things. What sort of things do we need to think about when we come up with a drug candidate are going to matter when we go and try and present this to a pharma company or a biotech company or a VC to bring more money in? What sort of things are they really thinking in the back of their mind that could trip them up? And the reason is because we operate in labs like this. I mean, I'm sure, walk around up here and this looks a bit like areas of my lab, and that's what we do. We sort of have our hood, we have our incubators, we have our flasks, and we do all of our experiments at this scale, and everything looks pretty good. We run a few gels, things look good. We tell people we've got stable stuff, that all looks good. So, what's the problem? Why can't we just turn that into a drug? Well, the reason is, and I don't think even I really fully appreciated this till I first walked into a commercial manufacturing facility. And if you ever get a chance to go to a large scale commercial manufacturing facility, I don't care if it's a small molecule facility or a cell therapy facility or a biotech facility, do it, because it'll change your whole mindset around what it takes to actually commercialize something. And so this is what lab at scale looks like. And so, you can sort of see the scale of the people here. You can see how many floors this is, and you can just imagine the type of rigors that something has to go through to be able to be manufactured at this scale, to hold up, whether it's just been held in a tank, whether it's been pumped around, whether it's been squirted into a vial, whatever it is, your product at some point, if it's going to be successful, has to survive this type of commercialization. And if it doesn't, no one's going to be interested in it, no matter how good the biology is. And I think that's a really difficult thing to hear, because sometimes the biology is really exciting. Right now, we're in a situation where we've got two types of advanced manufacturing for us in the biotech industry. One is this traditional bulk-based manufacturing, where we have these large centralized facilities. And this picture here, for example, actually is an investment that's happened in Korea. It's the Samsung facility there, if anyone's ever had a chance to see that. It's the largest biomanufacturing facility in the world. And down below is an example of where automation has taken us to put products into vials. So there's that type of facility, but there's also the on-demand patient-specific manufacturing, these paradigms that we can just make small batches of products for people because it's individualized to them or whatever the situation is. But both of these actually surprisingly have the same requirements in terms of the rigor that goes into them. And really, we're at a precipice for cell therapy, I think, over the next little while, of whether this on-demand manufacturing can actually make it from an economics point of view. So we're going to wait and see what happens there, because at the moment, that's a really challenging equation from an economics point of view. All right, so two important trends that I think you need to be aware of when you're thinking about developing products. One is that when we're developing products, one of the things we don't often think about is, what does it mean to the patient or the customer who will take my product at the end of the day? And I don't think the FDA ever really makes great slides, but I do like this one. I mean, it's pretty cartoony. But what it's saying is that whenever you think about making a product, you have to interlink three things outside of what you're doing in your lab. And that is, you need to think about how my product is going to be used by the patient at the end of the day, and how my product is going to be manufactured. And both those feedbacks are bidirectional, because if the patient at the end of the day requires something that is convenient to use, and they have to have it only dosed once every 6 months, then you better have a process in place to meet the needs of that patient. And actually, when you really start diving into it, there's a whole range of tools that people use to help inform those decisions. I don't have time to go into those, but you might have heard people talk about things like target product profiles or quality target product profiles. The point of those are actually checkbox exercises to make sure you're thinking about this. And so when you're thinking about both equality by design, making sure you design from the start, and also you're thinking about your patients at the end of the day, that you link these things together. The second is, how do you ensure that the products you make fit into a matrix in a competitive industry? And so you may not be the only one making your product. There might be 5 or 6 other people making the same sort of product as you. And so you need to be thinking about things in 3 ways. That is, how can you make it quickly? How can you make sure you've got a higher quality? How can you make sure you're doing it at economic scale that is competitive? And I don't have time to go into that, in any detail, but I will say that things that matter are things like target validation, how you deal with the data, talked about that before, how you do your clinical trials. You're going to do traditional clinical trials, you're going to think about new ways of doing things. But one thing that, I just put a box around it that I was involved with in the last sort of 10 years was thinking about moving to novel manufacturing paradigms. So you saw that facility that I showed you before in Korea. We built a facility in Singapore a few years ago as part of the company I was working for that was about 1/10 the size of that facility, but had a similar type of potential commercial output. So there are new ways of doing things as well. And I can talk about that if anyone's interested later. All right, so what are we all interested in as researchers? Whenever I have a meeting with a researcher about a product or something they want to do, usually it's a lot of slides about the biology, which is great. I love biology. Biology is really important. You have to actually engage your target. You have to make sure that you're actually interdicting a pathway of interest. But really, that's just one part of the equation. I think it's really important to think about what are the other things you should be thinking about. And so this is the way I like to think about early stage simplified product development. It doesn't need to be completely overwhelming, but you've got to make sure outside that circle of biology, you're thinking about the interconnection of these other things. So that is things like, what's my acceptable safety profile? How long does my molecule going to need to stick around in circulation? What's the impact of that on the bioactivity of the molecule? How does that impact my ability to manufacture? How do I then make sure that I can characterize that molecule and then control? And so Scott was asking before what CMC is, and that is chemistry manufacturing controls. That is making sure that that interconnectability between your ability to make it and your ability to characterize it, that you understand. And I think I just wanted to exemplify that. I guess everyone will recognize, say, why are you showing a boring picture of an antibody? Antibodies work because they've got CDRs and they bind targets and they're great molecules and they're really stable. And I've had one in my fridge that I've been using for the last 20 years and it still stains the same things and it never changes. Well, if you're talking about manufacturing a drug, things change a little bit. So when I think about an antibody, this is what keeps me up at night. It's like, oh my God, what things in this molecule are actually going to change? What is it about this particular interplay of these CDRs? How have they affected the physicochemical properties of this molecule? And what do I need to control from that? And so this is what I would say when, from an industry perspective, someone's thinking about when they're going to come in and characterize your molecule. So it comes back to that idea of, I've got a pure protein, for example, I ran it on a gel, I've got my single band, but actually what flows underneath that when you start to think about that from a commercialization or making a drug point of view is how are you going to characterize that? And there's a whole range of different ways that you need to be thinking about that, from your primary structure, higher order structures, what are you going to do from a pharmacokinetic point of view? So it's actually just some of these things. And if you want to, if you would like some references on this or to learn more about it, I'm happy to connect afterwards. Luckily, there are processes put in place that people do to really work through this in a stepwise fashion. And again, I've just put a reference up there. I won't go through it in detail, but there are ways you can de-risk these molecules and make sure you're doing it. And then there are also a really excellent set of tools that you can use to make sure you're thinking about all those right things to form those interconnected arrows between those areas to make sure that you can make good decisions early. And again, I can talk to people offline about that. So what's available to you as researchers to help with this? One of the things that we've been very fortunate to have funded over the last few years, although it has been a bumpy funding round, and we're hoping that through the next roadmap it won't be so bumpy, is a support across the country of a set of facilities that help researchers with this. And the facility that I'm involved with falls under Therapeutic Innovation Australia. Some of you may have heard of that. One of the nice things that TIA funds, it's 24 different facilities across the country that span small molecules, biologics and vaccines and cell and gene therapies. And the idea is that they try and support researchers through this. This is obviously a very expensive exercise. It hasn't been as well funded as it should have been prior to 2019. A lot of the facilities are rebuilding and trying to generate back to where they should be. But I think there's some promising times ahead. And so the idea is that these facilities, you can come, access them, get access to both the infrastructure and the expertise to go through from your early stage research screen to lead, go through your proof of concept studies, get advice on your preclinical strategy, and then move forward into your clinical manufacturing. Specifically for us, we call ourselves the National Biologics Facility. Again, it's trying to create that bridge through that early manufacturing through to development. And so trying to de-risk all those things that I've been talking about so that it really something that you can get access to from an expertise point of view to make sure you're making those right decisions. We're located across three major nodes across the country. So there's ourselves here at UQ, there's a training facility at University of Technology, Sydney, and then there's a facility down at CSIRO, which is going to do scale up and GMP. And so, we're hoping to build more aspects into this network, but that's where it is currently. If you look at that's just what I mentioned in terms of focus. So I think it's a really important part of our sovereign capability, and it's something that we've been talking extensively to government about. It's quite a difficult, complex story to tell, and really something that maybe in the US or elsewhere is filled just by contract manufacturing organizations or mid-sized biotech. That's something that we don't yet have here in any appreciable way in Australia, and we'd like to see more of it. So we're hoping this does spurn a bunch of other commercial operations. If you come over and look into my lab, this is what it sort of typically looks like. So we're fortunate to have some, pretty dedicated facilities to doing this type of work, both from a sort of small scale in that flask, as I talked about, but also going into sort of what small scale manufacturing looks like. So if you do need to transfer to a GMP facility, you can do that. More recently, we've been given a couple of $1 million to set up a pilot facility to support nucleic acid-based products. So we're calling that BASE. It doesn't stand for anything. It's just meant to be base as in base of a nucleic acid. And we're hoping that this facility will let researchers come have access to abilities to make things like plasmids, single-stranded DNA, or double-stranded DNA even, and also mRNA. So we've been given money to set that up. We're right in progress of getting in the capital equipment, hiring 5 new people to drive this. It'll be a collaboration between myself, Tim Mercer, and Linda Lua over there at AIBN. And so we're really, if you've got projects in the space that you think might need support for scale up, please come and talk to us as well. And just to say that, we've been fortunate to work with a large number of companies across the board. And I'll just say that there's so many great ideas out there. Since coming back a bit over a year and a half ago, it's been really fantastic to talk to people and companies about all the great things that are going on. We're hoping there can be some really great outcomes. All right. So I'm going to skip over a couple of things here. Just to say that I think, TMI as an announcement is so important from Brisbane, and we're really hoping that the rest of the money comes through because I think this is really where we need to be heading in thinking about how do we support that next stage of translation. Whether it's medical device manufacturing or whether it's more traditional manufacturing, it's really an area that I think if we can put more resources into, especially here in Queensland, we're going to see the fruits of that really soon. So I was really excited for that. All right, so where are we at for time? So I've got, I think, 10 minutes. I'm just going to give an update on our vaccine program. I know Paul talked about this extensively, so I'll just skip through to the important bits. So we all know that we've been talking about pandemics, and we didn't think it was going to happen. Fortunately, as Paul would have explained, there was an organization out there called CEPI who raised a couple of $2 billion to support research in this area. And they were working across a number of different disease areas, neglected tropical diseases like MERS, Lassa, Nipah, chikungunya, et cetera. But really importantly, they had this disease X paradigm where they decided to fund some platform technologies that could be used in case of a disease X outbreak. And as we all know, disease X happened. We're now all living with it in the form of SARS-CoV-2. And CEPI really played an important role in the solutions that have come forward. As you'll all remember, people were sort of saying, how fast can we go? What kind of vaccine can we develop for COVID? And if you haven't seen this picture, I think it's one of the most amazing things to come out of the last year and a half. And that really is our success rate and timelines for vaccine development. If you look all the way down the bottom, you can see a red bar and a green bar for our ability to bring forward a vaccine for COVID. And really, if you look at our history of dealing with infectious disease is quite astounding. And if you look at what CEPI funded, it's been really, fundamental. I mean, they funded programs at Novavax, at University of Oxford, at Moderna, at other programs you might not know so much about, like a Chinese company called Clover, or the European company CureVac, and as well as our own program. So very early on, providing large-scale support into this. As you'll all know as well, COVID has seen us throw the kitchen sink of vaccines at the disease. And for those who aren't aware, there's a whole range of different vaccine platforms. from genetic vaccines that have never been commercialized before that I'm sure many of you have already had. I know I've had Pfizer vaccine. I didn't think I'd be, having an mRNA-based vaccine as my solution to COVID, but there you go. To viral vector vaccines that, again, many of us are having here in this country, to more traditional approaches like protein vaccines, like the one we were trying to develop, or whole virus vaccines. And so I think I just wanted to make the point that the reason that those vaccines were able to move so fast is because there was new types of manufacturing, right? So if you haven't seen, there's going to be a documentary coming out, I think, on SBS that followed our program and a number of other programs around the country, around the world, but they followed the progress of Pfizer. And if you ever want to see what changing a manufacturing paradigm looks like and what it takes to actually throw resources behind something, what Pfizer's done and Moderna have done in the last year and a bit has been amazing. I think in the documentary, they say that Pfizer spent $800 million up front before even knowing what their candidate was going to be. It was a little bit different to the situation we had here in Queensland. But making protein vaccines is really, really difficult. And I'm sure Paul walked through the technology of the molecular clamp, which was the fundamental technology that the UQ team developed. And really what that technology does is it lets you take your viral surface glycoprotein and lock it into a pre-fusion conformation, which is essentially thought to be important for stimulating a robust immune response. And so the way that works is really, if you take the protein off the surface of the virus, it flips into its post-fusion conformation, which is its extended conformation, which is what you don't want from an immune recognition point of view. What you really want is this globular compact pre-fusion conformation, and appending the clamp on here is able to lock that trimer into that pre-fusion conformation. The clamp that we used, as you all know, was a trimerization domain derived from HOV protein GP41, and that becomes important for what I'm going to tell you. And the team worked really hard at showing over a number of years that this technology worked on a whole range of different diseases. And so that let us bring in around $10 million of funding from CEPI to develop a platform from going from DNA sequence through to vaccine candidate as quickly as possible using this clamp platform. And so last year, we did that for COVID. We had to think of really different ways of doing things and moving really fast. So I'm not going to spend any time on that. But what was the really important thing? Well, the really important thing is we had to think about going from our flask, our 30 ml flask that we were doing in the lab, through to transferring something that was going to be acceptable from a global commercial supply point of view. And so that was really the big challenge for us. I knew it was the number one thing we started thinking about right at the beginning of this program. So how did we do that? I think, as you've heard, one of the really fortunate things we have, although we don't have a huge biotech sector here in Australia, we are very fortunate to have presence of CSL Seqirus here. And it just so happened that they had exactly the right manufacturing platform to try and do manufacturing for us. And so we were able to do a really intense handoff with them straight through our preclinical and early preclinical. So they were able to start commercial manufacturing in Q3 last year. And so that was a really incredible effort. I think, it's not talked about very much, but they were able to run multiple commercial scale GMP runs of our vaccine candidate, make millions of potential doses frozen away down there at risk while we were going on with our program. And I think why proteins are so important, as you will have seen if you've been following the vaccine story closely, is because they have a much more maligned safety profile than other vaccines, especially in terms of tolerability. So this is some data from our clinical trial. And basically, most of the reactions, except for injection site pain, were indistinguishable from placebo. That's quite different from the viral vector, or if you've had your mRNA vaccines. I know I had a pretty solid reaction to my dose 2 for Pfizer, and that generally doesn't happen for these adjuvanted protein-based vaccines. Really why we were pushing so hard. Just to say, did the vaccine work? We'll never know, because as you know, I'll tell you, it's not going into phase 3 studies. What we do know is that both across younger adults and older adults, we were able to stimulate a very robust immune response. We couldn't have hoped for a better outcome. And as some of you may know, people have been looking now at correlates of protection and really looking at how well these vaccines would have worked. And this is from a number of our Australian collaborators looking at plotting that immune response and against efficacy. And if we plot our results right on top of that, we fall right at the top of the curve. So we felt pretty good that had we had the chance to test efficacy, we would have been right up there in terms of a protein-based vaccine. And we'll see how that plays out for Novavax. Hopefully they get approved. And so we all followed this coronavirus tracker last year, and I know I did. I think New York Times had one of the best graphics departments working on this stuff and kept it really updated. But the one, of course, sad day for us was when we came in and we saw there was a new category added here, and that was the red one that we don't like. And that was the abandoned vaccines. And we're in pretty good company here with other groups who decided not to progress their vaccine. And I'll tell you the reason for that in a minute. But the one thing I'll say from a translation point of view is that we really compressed many years of work into really just a few months. And so within 12 months, we checked through every technical barrier we could imagine, proceeded into commercial manufacturing, and we're really ready to go in that last week of December for filing our phase 3 protocols in, I think we're going into 22 different jurisdictions and something like 170 clinical sites prior to the decision not to move ahead. And just to say, the reason for not moving ahead is one of those things where you really need to think about your program early. And that's, I mentioned, the trimerization domain we used for this vaccine was derived from HOV GP41. There are many reasons for that, and I could talk about another time, but it really just works really well. And it also let us do certain elements of our manufacturing that we couldn't have done in such a speed any other way. But the downside of that really was that that domain not only worked well from a stabilization point of view, but it also generated an immune response. And if we have a look at what that immune response did, it generated specific antibodies against GP41, which are used in a number of HOV diagnostic tests. Not all, but a number, and a significant number, especially some of the big lab-based assays. And that meant that folks who have had the UQ vaccine would have come up as a false positive on some of those HOV tests. But I think what's really interesting to look at is actually what was the level of that response. I'll see if I can mark it out here. So this is the placebo. These are the levels of the GP41 antibodies across some different dose groups that we looked at. And over here, this is what the typical positive response looked like. And this is on a log graph from HOV positive individuals. So you see the vast majority are way up here in terms of cross-reactivity. There are some very low. All of ours were really right down here on the bottom. But the key thing is we couldn't easily distinguish those folks who are false positive from those folks who are potentially HOV positive. And of course, I think the big question to ask is, why are we still doing this type of diagnostic testing for HOV when potentially molecular testing might be a better way to go? But really thinking about changing the face of HOV diagnostics was just not an option, especially in areas of the world where this disease continues to be out of control. I mean, for example, in Brazil, as far as I understand, you can go in, get one of these point of care diagnostic tests. If you come up positive, they'll put you straight on antivirals. And so obviously, if that was a vaccine-induced false positive, that's not something you would want to be happening. All right, so that was a bit of a whistle stop, and I think I've got a few minutes left for questions, and I'll finish up, and thanks online and in the room for listening.
Thanks very much, Trent. Great talk. We do have some time for some questions. Alan, let me just see what we've got here. So, have you got any questions in the room? John, and then Frank.
Yeah. Sure. Yeah, so I'll just repeat the question. So John Hooper asked the question in terms of halting the UQ vaccine program, how was that decision made? It's a pretty complicated answer to that, but basically the Australian government has scientific advisory groups that they rely on for advice in making those decisions. And so it's one of those groups that decided unanimously that the risk to both vaccine hesitancy and potential disruption of HIV diagnostics was just too great at that time. And it came, I think it's important for context, came right at the time where we started to have positive data both for the AstraZeneca and Pfizer programs. So we knew other solutions were available. Prior to that, we weren't really sure if, how these efficacy data were going to read out. So things may have been different had those efficacy readouts come at a different time.
Right.
Yeah, sure. So I think Frank's asking a question of, we seem to get stuck in a rock and a hard place both times. We get pinged because, we do lots of basic research with focusing on publications and not translational outcomes. And then at the same time, industry is getting financially sort of impacted by their ability to not turn R&D into translation as well, commercial products as well. So I don't have a good answer for that. My general answer is that I don't think we should fund translational research. I think we should just fund research in general. And I think it's the base funding that really matters. I, like many people, spent time in the UK and was fortunate to be supported by groups like Wellcome Trust and Cancer Research UK. And there, I don't ever remember a question of why my Drosophila genetics was going to lead to a drug or not lead to a drug, but, as in one of the most well-funded groups in the UK. And so I think that's where we really need to be educating our funders and politicians on the importance of funding high quality research and keeping a dynamic community alive. So that's a bit of a cop-out, but I don't have a better answer than that.
Thanks, Trent. So we've got a couple of questions here about the new entrepreneurial PhD program, firstly from Fernando and then from Mahir Gandhi. Is there an application scheme or guideline? Is an AIBN co-supervisor required? And are PhD students currently on a conventional program able to enroll in the program? That's probably a good starting point. Yep. And then I'll come back with some more.
Yeah, sure. So great question. So yes, my understanding is I believe you do have to have a supervisor in AIBN because AIBN is putting quite considerable funding into supporting the program. So I believe that's a requirement. It's also a new scheme. So I think the goal is to enroll in the entrepreneurial PhD program from the beginning. But what I would suggest, if you're able to email me, I'll put you in touch with our HDR person at AIBN, and they can provide further details just to make sure I'm giving accurate information because I don't want to get into trouble.
Thanks, Trent. And also, Fernando, I should highlight you made the comment of a great talk. And Mahir Gandhi's also asking the question, how do you find out more? Well, I think that's now answered in your questions. And then any more questions? Yes, Norbert.
Yeah, that's a great question. So the question was, what's Australia doing to boost ability to make more RNA-based products here in Australia if that becomes a major technology driver? So the answer is complex. So right now, the federal government's put forward what's called an approach to market, a call for an approach to market. And so there's a whole, it closes in a week, I think, closes on the 16th of July, where companies have been asked to put proposals forward for how they can bring RNA manufacturing capability here to Australia. And so the important part of that, there's two important parts. One is they need to be able to bring the IP to be able to do that. And the second part is they need to be able to bring a strategy of how they're going to engage with the research community. And so I think that's really, really important because just having a commercial facility here will not benefit the researchers. But if they're going to bring both a commercial facility and R&D capability, that will benefit researchers. So that's the strategy that they're going for. We don't know how long that's going to take or how it's going to play out. I can say I've spoken to quite a few of the companies who are putting proposals into that, and I've been really buoyed by their desire to engage with the research community. And I think it is just way beyond infectious disease. I mean, as you say, if this becomes a way of doing, transient gene replacement, or there are other types of disease or therapeutic areas that now the ice has been broken for, we'll see. But it's a big challenge. RNA's had a long checkered history, and we've been very successful here. I don't think it's going to be a one size fits all solution, but I think we have to be ready to play in that space. Our pilot investment here, we're hoping that leads to a whole network of facilities across the country. The Australian Academy of Science is organising a roundtable on the 29th of July, which I'm hoping is going to be broadcast. And so the few of us are trying to work out what we're going to do for that roundtable. It's sort of Australia's first scientific discussion. And there's the Australian RNA Production Consortium. It's led by a bunch of researchers across the country, including people like Damien Purcell, Colin Putin, Nigel McMillan, Archer Fox, and others who are looking at using that as an opportunity to talk more about this in the Australian ecosystem. Yeah, I don't know. I'll send you the, there's a great, I've been reading a lot of New York Times, obviously. Great New York Times article on what Pfizer's manufacturing facility for the COVID vaccine. Maybe you can just Google it as well. And that really shows you the scale of what it takes to even do, and vaccines, low dose, but what they had to do from an industrialization point of view to take in vitro translation, in vitro transcription to a commercial scale. I mean, it's really kind of mind-blowing. So, I think it still has a point of relevance.
So one of the terms we've heard a lot about was sovereign capability in the last 12 months. Do you think in a sustainable way, bringing more high-tech, med-tech industry on board should be with large pharma bringing branches here, or should we be trying to somehow develop our own industry, which may flounder in the future outside a pandemic?
Yeah. So I think both. Actually, when I used to come, I worked for a biotech company before, and when I came back to Australia to do some work for the Australian affiliate, I was really disheartened because the affiliate, had government relations and sales and marketing, and that's the case for the vast majority of biotech who has presence here in Australia. And I think we've got to find a way of encouraging those companies to bring branches of their R&D here, whether it's immunology, whether it's oncology, whatever it is. We've got to get them to come and bring some of those divisions here. And I think that's really what's needed to then build confidence in what's possible here in Australia. Somewhat controversially, I even don't really love some of the, innovation centers and things that the big companies do here, because really that's just about harvesting IP as early as possible and taking it back to the mothership. I don't think that benefits as well as it could either. Maybe that's controversial to say, but I think in partnership it can. If we can show what's possible here, and there's good connectivity back, and then that leads to labs being set up and staff being here from those companies. I think that's important. And the other thing is that there's no doubt that mid-tier biotech is the driver of innovation. So startups are great, but it's really that mid-tier that end up having an M&A strategy with a large entity, which changes the needle in terms of having then a sustainable biotech ecosystem.
So the last theme of questions are online, and they're both related to the UQ clamp technology. The first was, let me just go back to the top. With the development of the HIV-based clamp technology, could it have been predicted earlier that this may interfere with testing? Maybe you want to move on fairly quickly after that question to saying, are you still working on the molecular clamp technology, and how do you want to apply it now, given what you know about HIV?
Yep, perfect. Yeah, so we recognized that it was a risk, and it was something that we flagged early on, that potentially antibodies against that trimerization domain may interfere with some diagnostic assays. What we knew at the time is actually the domain was in its post-fusion conformation in the way that we use it. And so we expected that the majority of antibodies that were generated against that domain wouldn't cross-react with the sort of native protein that's used in diagnostics, or the peptides that are used in some of the diagnostics. And actually that turns out to be the case. So it's only some forms of those diagnostic assays and some of the GP41 antigens that are actually cross-reactive. There wasn't a way that we could have easily predicted that without having human antibodies. And as soon as we could predict it is when we found out about it. So that's the reason. So we've been trying to, I know this is being recorded, but we've been trying to stay under the radar a little bit in terms of what we're doing now. But the team has been really hard at work, especially Paul Young and Keith Chappell and Dan Waterson's teams on developing a clamp 2.0. So that's going well. They're using a new trimerization domain from a different virus, not a human virus, not something that could interfere with any diagnostics. And that work's progressing really well. And we're continuing to work with CEPI and our funders on that. And we're expecting to move as quickly as we can. We think, different types of boosters for COVID and different types of defenses for future pandemics are critical. And we're continuing to push as fast as we can on that.
Thanks, Trent. I think we'll have to wind up the session. I think you'd agree with me. It's been really amazing to hear about your experience, but also your wisdom from working in biotech. It's great to have you back in the country. And with that, can we just thank Trent for travelling across the river to talk to us today?